Well, we just have a spoonful of scientific research here in this session with 2 speakers only, but these topics are really important. So the moderator is Dr. Indu Kaur of India, and she'll be speaking first on controlling intraocular pressure with novel, patented, nano-designed implants and gels. Second is mitochondrial delivery of RNA and DNA towards mitochondrial gene therapy of optic neuropathies by Dr. Volker Patzel, who's based in Singapore. We're fascinated by these scientific topics, so we wanted to give them plenty of space for you to digest. Enjoy.
So, hi everybody. So we are here for the first session of track 2 today for this Cake and Pie Expo, which is a fascinating way the organizers are trying to present the scientific content. So I'm here as the moderator for this session, and we have the first talk by myself, and I would just briefly try and introduce myself, and then I would also introduce the second speaker for today, that is Dr. Patzel, who's also with us here, and then we'll start with the talks. So I am Indu Kaur. I'm a professor and chairperson at University Institute of Pharmaceutical Sciences, Punjab University, Chandigarh, India. So previous to that, I was director of the Sophisticated Analytical Instrumentation Facility at my university, and then the dean of the Faculty of Pharmaceutical Sciences. I am amongst the top 2% most cited scientists in the area of pharmacy and pharmacology across the globe, as promulgated by a scientist from Stanford University, USA, last year. My research forte is enhancing the bioperformance of drugs and biomolecules using active tailored delivery systems like solid lipid nanoparticles, nanovesicular systems, and cyclodextrin complexes. We've been working with we've been trying to develop some novel platform technologies for overcoming challenges in ocular delivery of a variety of molecules. 2 of them I'll be discussing today, that is atorvastatin and bimatoprost. Then we worked with acetazolamide, some antifungals like ketoconazole and fluconazole, and timolol and natamycin. I have 10 Indian and 1 US patent granted to me, and have filed 16 more patents over the past 10 years. Out of these 11 granted patents, 6 are in ocular delivery. I have mentored almost 25 PhD graduates, and I was a US Fulbright Fellow for the session 2017-18. I was also awarded the Women Best Scientist Award in 2018 by the Organization of Pharmaceutical Producers of India. I have several publications to my credit, and I have also been graced with some technology exposition awards over the past several years. So that was a brief about me, and I'll be talking about 2 of my patented technologies today for the management of glaucoma. And we are also very fortunate to have with us Dr. Volker Patzel from Germany. He's a chemist who graduated from the Institute for Biochemistry at Technical University of Darmstadt. Please forgive me if I do not pronounce it correctly. So Darmstadt, Germany, and he received his PhD from Ruprecht Karls University in Heidelberg, and subsequently he did a MBA from Steinbeis University in Berlin. He worked as a postdoctoral fellow at German Cancer Research Center in Heidelberg, and then as the research group leader at the Max Planck Institute for Infection Biology in Berlin. He joined the National University of Singapore in 2009 with a dual appointment as assistant professor at the university, and also the assistant director of research at the University of Cambridge, UK. Currently, he's a senior lecturer at National University of Singapore. He has 13 patents and applications and more than 50 publications to his credit. He's the founder of the Steinbeis Transfer Center for Nucleic Acids Design in Stuttgart, Germany, and of Avacrec Pte. Limited in Singapore. His research focus is on the development of RNA-based genetic therapies, and today he would be talking about the mitochondrial gene therapy for the damage of the optic nerve. So with this, I think we will start with the first presentation, and you're welcome to ask any questions during the talk. And we can also have a QA session at the end. So whichever way the audience wants, we are game for it. So I hope you can see my screen. And the topic for my talk today is controlling intraocular pressure using novel patented nano-designed implants and gel. So this is a picture about my university. This is one of the landmarks in Punjab University. And here I show the one of the emblems of my city, Chandigarh, which is an open hand that we welcome all cultures, newer thoughts, and newer opinions. So my university is amongst one of the oldest in India. We are also at the top. We have almost 550 acres of land, 78 teaching departments and centers. So this is my institute where I work. This is the front building, and this is a building at the back of this building. So I am very proud of my institute. We are almost 76 years old Institute of Pharmaceutical Sciences, which is numbered 2 in my country. We have to our credit several new molecules which have been discovered, one drug. So from a state university, I would say a public funded university, producing a drug is a great thing. And we have more than 10 technologies which have been transferred to the industry from the institute, and the good citations and good number of publications. And 8 of our faculty are among the top 2% in the world in the area of pharmacy and pharmacology. So I've just structured my talk around the issues with the conventional delivery and how the novel nanotechnology systems can improve this, and a brief about what I have done and the type of results I got. So when we talk of the normal, ocular drops, suspensions, or ointments, at the face of it, it is said that less than 1% of what is delivered into the eye is absorbed and elicits the physiological effect. Unfortunately, developing drugs for ocular effects, for ocular delivery has not been, it is usually seen that the drugs were developed for some other applications, and subsequently they found their application for ocular delivery. As a result, the molecules have never been tailored to deliver across the eye, where the issues or challenges are like, you have an epithelium, you have the cornea, which is a 5-layered tissue with and broadly speaking, we talk of 3 layers. You have the outermost corneal epithelium, the inner endothelium, and in between a stroma. So both the epithelium and endothelium have a lipophilic nature, while the stroma is aqueous. So you have to have molecule which has both hydrophilic and lipophilic character, which is very difficult. Usually you have lipophilic molecules, and at the same time you need to deliver it in the form of ocular drops, so it should have some inherent solubility as well. So mostly drugs are lipophilic in nature, and delivering them as ocular drops is an issue. In addition to that, blinking, tearing, once you instill something into the eye as a protective mechanism, leads to dilution of the drug solution, and then spillage on the cheek leads to losses. Further to that, some amount of the solution pours into the systemic circulation via the nasolacrimal duct. So there is again loss, and then there could be systemic side effects. And further to that, the residence time of any instilled solution in the eye is very low. So what is usually done is, especially for chronic ailments, the patient is advised to frequently administer the drops at least 3 times a day. Many a times, for antifungal drugs or other such drugs, the administration time can be as fast as every 4 hours. Then, because you are administering frequently, so chances of nasolacrimal drainage and then subsequent systemic toxicity are high, and then irritability could be an issue. Then since it is a multi-dose system, so you add preservatives into it, which can cause irritation. Then the answer could be ointments, where we could have a longer residence time in the eye, but most of the ointments, they produce blurred vision. So usually they are recommended for nighttime use, and the partitioning of the drug from the ointment base towards the eye needs to be considered. You have suspensions, which are particulate systems, and the particles, they are said to lie within the cul-de-sac, and as a result produce a longer residence. But compliance, the stability issues, the release of the drug from the suspended particles, all of them have to be considered in formulation design. So what I propose is a nanocolloidal system, which would give you nanoparticles. As a result, there would be an improved permeation. Then the drug is released slowly, so you have a longer duration of action. And since the particles would remain residing in the eye and also permeate across the cornea because of their nano nature, so systemic losses would be minimum and side effects would be less. And as a result, you can have once a day installation advised to the patient. And if you have developed implants, then you can have even a longer effect of up to several weeks and months. And some in situ gelling mucoadhesive systems have also been proposed. One of them is Optic, which was launched by Merck. So one system which I want to discuss here, which is novel, is a nanovesicular system, but it is different from the conventional liposomal system. And why I wanted to do that was that liposomes usually involve a phospholipid and a cholesterol. And both of these molecules are biological in nature, which makes them really versatile. And that is what is attractive about them. But at the same time, they are prone to oxidation and hydrolysis, which leads to the compromised stability of the system. Then since they are natural molecules, so they are costly. And since, they could degrade, so there could be a leakiness from the liposomal system. And as a result, there is batch to batch variation because of the natural origin, and the methods of manufacture which are available are not really amenable to industrial manufacturing, and the reproducibility is also found to be low. So what I proposed here was using some synthetic polymers and surfactants. We prepared a vesicular system. The components which were selected are easy to procure, less costly, and since they are synthetic in nature, so batch to batch variations are not there, and it can be, and the method of manufacture was very simple. You just, mix the two. You have an organic phase and you have an aqueous phase, and you just mix them in a stream and you get vesicles which are dispersed in an aqueous system. So this is the picture of the vesicular system I have developed. It is a multilamellar structure, and The drug we selected was the metoprolol, which is a well-tolerated, stable prostaglandin of choice for glaucoma. Issues with the drug are very poor solubility. It is lipophilic, which is again favorable, but it has a large molecular weight, so may show a poor is not a very big issue in the sense that you need a very small concentration of the drug in the drops for effectiveness, and the IOP is very effectively lowered. But you have to repeatedly instill. As a result, patient adherence is poor. Further to that, benzalkonium chloride is added as a preservative into these drops, and repetitive dosing leads to itching, burning, and dry eye type of an issue. So when I discussed with some physicians in and around my institute, I found that they are looking at an option where they can have an implant which would be effective for up to 3 months or so, or a once a week drop. So that is what gave me an idea. So this is the system I developed, and the beauty of the system is that it is autoclavable, because in the eye you want the solution to be sterile, and sterilization is could be an issue with some of these nano systems. By autoclaving, you have to then manufacture them aseptically, which is a costly process. So we prepared these nanovesicles and extensively characterized them for ocular suitability in terms of their pH, their osmotic pressure, their viscosity, their plasticity, their sterility, and their suitability for employment of a preservative into them. And then we converted these nanovesicular aqueous dispersion into an in situ gelling drops. So what do you mean by in situ gelling is that you have a polymer which goes into solution, but it is liquid at the temperature of use. And when you instill it into the eye, or for that matter any other tissue, due to certain changes, that could be pH, certain triggers like a pH, a chemical reaction, or an enzymatic reaction, or some ionic reaction, the polymer would gel. And as it gels, so it retains the drug and releases it slowly, and you get an enhanced effect. So what we did was we clubbed two systems. We have a nanovesicular system, which would ensure a better permeation and also a controlled release. And then we further incorporated it into a in situ gelling system, which would further improve its release and delay and improve its stay in the eye. So we evaluated its safety, stability, and then, as I said, autoclavability. It showed an extended release for up to 10 days, and the intraocular pressure was lowered in the rats, and it extended up to 5 days after installation of a single drop. So you can have a once a week installation, because glaucoma is a disease which is, chronic, and the patient has to use medication throughout life. So it can be a great help if the patient can instill it just once a day. Or we also evaluated it as a subconjunctival implant, where in preclinically we could show the effectiveness up to 2 months, and clinical effects may further extend the stay. So this is how you see my vesicles under a transmission electron microscope. You have an outer surfactant layer and a bilamellar structure of the vesicles. So this is how I, furthered the dispersion. We added some polymers into it, and you can see that it is a liquid under refrigeration or at room temperature. And if you incubate it at 37 degrees, so it is a temperature sensitive gel which gels at 37 degrees. Further to that, it showed a good shear thinning. That means when you instill into the eye, it would not cause any irritation and would spread across the eye uniformly. So if I, try and represent it schematically. So as I said, you have an organic phase and an aqueous phase in which you have added the drug. And when you mix the aqueous phase to the organic phase, you end up getting nano vesicles, which encapsulate dermatoprost. And then you further add gelling agent to it, and you get a liquid, which when instilled into the rabbit eye gels. So you have this gel matrix, which has nanoparticles embedded in it. And so what would happen is, one is the gel matrix would dissolve, and then it would, you have, as I said, 88% of the drug was encapsulated. So almost 12% was lying free in the solution. So at the initial phase, this free drug, which is in the gel matrix, would be released. And then along with that, the particles also start releasing the drug, and you get an extended effect. So then we evaluated the intraocular pressure lowering effects in rats. So first we induced glaucoma in the animals by instilling steroid 4 times a day for almost 4 weeks. And then from 4 weeks onwards, we started instilling dexamethasone twice a day. And at this, after 4 weeks, when we were sure that the glaucoma, the increase in intraocular pressure has been induced, then we divided our animals into 7 groups. We had a naive control group, which was not treated by dexamethasone. Then we had a positive control group, which was not exposed to any treatment modality. Then we compared it with Lumigan, which is a marketed formulation of bematoprost, and we would administer Lumigan every day. This is a formulation which is recommended once a day. We employed 2 vehicle control groups, one for the drops and one for the subconjunctival implant. And then we had 2 treatment groups. And this is the type of results we got. This line shows the results for the marketed formulation, which was instilled every day. And this is for the subconjunctival implant, which was injected below the conjunctiva. And we could see the reduction in IOP for almost 6 weeks or so. And this is again a drop of the marketed formulation and a drop of our in situ gelling drops. And you can see that the effect lasted for almost 5 days. Then we evaluated the stability of the system, both in terms of the drug assay, the encapsulation efficiency, and the particle size upon autoclaving, and also for different, for up to 3 months at 37 degrees and also under refrigeration. And in each of the case, the results were quite promising. We also evaluated because it's a nano system, so you want to confirm that it is safe. It would not induce any, irritation or any toxic effects for your ocular tissue. So we took 3 cell lines: the corneal cell line, the conjunctival cell line, and the retinal cell line. And the product was found to be safe at several doses which were evaluated. Then we also performed preclinical safety of the product, and again we got favorable results. This slide shows the uptake of what we did. Was we labeled our vesicular system with a fluorescent dye, and we checked the uptake in the corneal cells and the conjunctival cells, and we saw that the uptake into these cells was significantly high, almost 25 times high. Then similarly, we also instilled it into the rat eye, and we sacrificed the animals, cut histological sections, and observed them under the confocal microscope. And again, we observed that the subconjunctival implant, we could observe fluorescence up to 2 months. And in case of the single drop, we could see the fluorescence up to 7 days. Then the second system I would briefly touch upon here is the solid lipid nanoparticulate system. So basically what it is if I compare it with an emulsion system where you have an oil and water system, so instead of the oil, you have a high melting point lipid, which is solid at room temperature. And because of being a solid matrix, you get a controlled release of the drug. So it compares well with the polymeric system, but it overcomes the issues with polymeric systems because of the toxicity, biocompatibility, poor manufacturability, poor drug load, and things like that, and the cost. So all of them are addressed here. And then vis-a-vis a liposomal system where you have a mobile core. Here you have a solid matrix, and as a result, you get a better controlled release and a more stable system. Then in addition to that, these lipidic nanoparticles are known to interact with the glycoproteins of cornea and conjunctiva so that they act, they form a depot and the drug is released slowly. They also interact with the lipid layers of the tear film such that they have a longer stay in the eye. So what we did here was we developed an ocular formulation for atorvastatin. Statins are pleiotropic molecules originally launched for the cholesterol lowering effect, but at the same time, they are now being explored for a variety of ocular ailments, right from diabetic retinopathy to age-related macular degeneration, and there are some indications that they could be used against glaucomatous damage in the retinal ganglion cells because of the inhibition of isoprenylation of Rho GTPase. Normally, if you administer statins orally, the amount of statin which can enter into the ocular tissue following systemic administration would be very low. And if the effective concentration is not achieved, you may not see the effects. And for ocular administration, there are further challenges, like it is insoluble. You would have to circumvent the various barriers, and it has a high molecular weight. In addition to that, it is an unstable molecule which is prone to even photodegradation and also sensitive to various affectants. So developing it as a nano system could be an issue. So what we did was, again, this is an autoclavable system, as you need a sterile product for ocular delivery. So we produced an optimized formula. The method we used was high pressure homogenization, which is an industry amenable process. So we could achieve high concentrations of atorvastatin both in the aqueous humor almost 8 times and in the vitreous almost 12 times. We established its safety, stability, and it could be an option for non-invasive treatment of glaucoma as well as other posterior eye diseases. So this is a schematic I tried to show where statin could be useful for a wide variety of ocular ailments, but it has a poor oral bioavailability, and as a result of which actual availability in the ocular tissue could be further limited. So if we develop a local system, it would be an answer. So this is the optimization we did to get these solid lipid nanoparticles. This is the transmission electrode. This is the particle size was around 250 to 300 nanometers. We got an extended release. It could permeate the cornea. We established the safety and the stability, and these are the ocular kinetic studies and also cellular uptake into the retinal cells. So with this, I come to the end of my talk. And as a scientist, I think what we need is to have a good workspace, an innovative idea to work around, the passion and the faith in the concept, some hard work, teamwork. I have a very good group of PhD students working with me, and I would like to make a difference. So thank you. And I am open for any questions at this. I see some question answers here. So I will just try and quickly. Okay, so this is a question by Gloria, and she says, how soon do you think will we see this nanomaterial in products out in the market. See, Gloria, I'm very hopeful. I'm looking for industry partners, and I'm trying to pitch in my technology since the patent has also been granted, and let's hope it can see the light of the day. So I'm quite hopeful, but I can't give you exact timelines in the sense that first is that I find a partner, then second is we will have to do some clinical studies, and then there could be some regulatory issues. So maybe 2 to 3 years if I find a partner soon. Okay, so there is Bala Vikash, and he says that, is it possible to make liposomes to target posterior globe? Yes, there have been studies where people have shown that liposomes can deliver the drug to the posterior eye, and some very favorable results, but I would still, try and convey that you have better systems which could be more translatable to the industry and which could give you better results.
Yeah, first of all, thanks to the Media Mice and the Cake and the Pie team for inviting me, and thank you, Dr. Kaur, for the kind introduction. So I'm talking today about a novel mitochondrial delivery vector system for RNA and DNA, which we are exploring towards mitochondrial gene therapy. First of all, of optic neuropathies. So as mentioned, my name is Volker Patzel, and I'm working at the Department of Microbiology and Immunology at the National University in Singapore. So if we have a look at the cuts through a human cell here in the center with the nucleus, we always have to be aware that we have our cells harbor two types of DNA genomes. First of all, the nuclear DNA found in the nucleus, double-stranded DNA organized in chromosomes, more than 3 billion base pairs, and comprising about 47,000 genes. The majority of them are non-coding. On the other hand, we have a second genome, which is fully maternally inherited and found in the mitochondria. It's only about 16,000 base pairs. We have about 2 to 10 copies of this genome in each of our mitochondria. And these are encoding 37 genes in total. Only 13 are peptide coding genes. And so that's why the majority of the proteins, which are important for mitochondrial function, are indeed encoded in the nucleus. And about almost 5,000 proteins encoded by the nucleus are synthesized in the cytoplasm and delivered via leader peptides into the mitochondria to function. And the mitochondria, as you all know, are double membrane bound organelles found in most eukaryotic cells, and they are very important for our ATP synthesis. They play also a role in aging and inflammation and in cancer. Looking at the DNA damage and repair mechanisms, well, for the nuclear DNA, we have very efficient, sophisticated repair mechanisms, both for single-stranded and double-stranded DNA repair. For the double-stranded DNA repair, it's I just want to highlight non-homologous end joining and homology-directed repair. On the other hand, it's a bit different for our mitochondrial DNA, which can also be affected by inherited and acquired mutations. But here, the repair mechanisms are less robust, and so we have here the oxidative phosphorylation, which creates an oxidative environment in the mitochondria, and the mitochondrial DNA, the consequence, is exposed to reactive oxidant species and free radicals. That's why with the aging process, we are accumulating defects in our mitochondrial DNA. And if these defects reach a certain threshold, let's say more than 80% of our mitochondrial DNA genomes are defect, then the cell function is impaired. And this accumulation of defects is happening with the aging process. So mitochondrial diseases can then also be associated either with nuclear DNA defects or with mitochondrial DNA defects. I listed here a couple of diseases which are associated with nuclear DNA defects, and on the right side, those associated with mitochondrial DNA defects. Indeed, defects in all of the mitochondrial genes have been associated with mitochondrial disease in humans, and I just give you here a small list for your consideration. So how can we therapy now mitochondrial diseases, and is there any gene therapy around? Well, I mean, gene therapy is still in its very early stage, and it makes again a difference whether the defect is associated with DNA, with nuclear DNA, or with mitochondrial DNA. So for the nuclear DNA, since a while, people managed to deliver healthy or functional genes using liposome and RNA carriers, non-viral vectors or viral vectors into the cells, into the target cells. This DNA has to enter the nucleus, and then it can be the genes can be expressed. So there will be proteins synthesized in the cytoplasm, and with the leader peptide, people are able to target them into the mitochondria. This works to some extent, and there have been first on a limited number of clinical trials documented. More recently, we also know that there is a CRISPR-Cas technology around, which might also enable us to repair defects in the genomic DNA directly in the near future. On the other hand, the mitochondrial DNA, the mitochondria are more difficult to access. There are two systems reported which can deliver RNA, but rather inefficiently into the mitochondria. This is mTRES and the mitoporter system. And also very recently, last year, the split DDDA technology was reported, which enables editing of mitochondrial DNA, but only cytosine basis. And it does not involve any nucleic acid delivery. So here again, a protein synthesized in the cytoplasm is being delivered in the mitochondria. So to summarize, about 1 in 5,000 individuals are affected by mitochondrial diseases, predominantly affecting the brain, muscle, the heart, and the eyes, or the energy-intensive organs. And mitochondrial gene therapy could provide a cure or treatment, but it doesn't exist yet, really, due to the lack of an efficient mitochondrial delivery vector system. So our target to investigate our mitochondrial delivery vector system is Leber's hereditary optic neuropathy, or LON, as a proof of concept study. LON is an orphan disease which causes sudden blindness in young adults, and it goes back to mutations in 3 of the mitochondrial genes, ND1, 4, and 6. The eye is a very suitable organ for targeting by nucleic acids. It's immune privileged, and it's accessible via direct injection. And for this disease here, there are well-phenotyped cell lines available, as well as a mouse model, which is quite unique for mitochondrial disease. And important also, the current treatment options have not met primary endpoints in clinical trials. So we aim to now provide a solution. We want to developing a mitochondrial gene therapy platform targeting LON disease and possibly other diseases. We hope to prevent and restore vision loss. Our project is at an exploratory stage at the moment. We have high fidelity prototypes, which together with some supporting elements will be tested in a simulated environment. It's a platform technology which allows us to deliver both RNA, non-coding or coding, and DNA all together. So it means basically any technology into the mitochondria. And we are exploring this technology for mitochondrial gene therapy towards human diseases, including cancer, controlling inflammation and immunity, and possibly also for anti-aging. So how does the technology look like? Mitochondrial gene therapy, of course, requires delivery of nucleic acids into the mitochondria. And the mitochondrial double membrane has been an unbreakable hurdle so far for nucleic acids. But as mentioned, we developed an efficient, modular, and scalable mitochondrial delivery vector for both RNA or DNA. It's based on a long non-coding RNA originating from the human cytomegalovirus, which has a length of 2,500 nucleotides. It's indeed the most abundant RNA produced by this virus during a viral infection. And this was found to localize to the mitochondria and to bind and stabilize complex 1. We have intensively studied this RNA and by computational means identified 4 RNA subdomains called D1 to D4, which are governing mitochondrial delivery. And we can see here, as compared with the activity, mitochondrial targeting activity of the full-length beta 2.7 RNA, the best domain seems to be with the highest overall activity, domain 3, which is as good or slightly better. And the one domain number 2 here looks a bit less efficient, but it's indeed the most efficient compared to the length. So the highest efficiency per nucleotide. So we first studied and tested whether we can use the original beta 2.7 RNA and fuse it to messenger RNA coding for GFP, and whether this is enabling recombinant GFP expression in the mitochondria. And indeed, it worked. You can see the mitochondrial gene expression here. So this was a messenger RNA for coding for GFP, which cannot be translated in the cytoplasm. it can only be translated in the mitochondria due to the alternative codon usage. It's not a very strong expression, as you can see, but there was also a premature stop codon in the sequence. That's why the GFP here is not expression is not very profound. The effects become much clearer when we now use a new repeat arrangement. So we here take 8 of our identified domains in a certain order and fuse it to an antisense RNA targeting the mitochondrial ATP6 gene. And this resulted then in a knockdown on the protein level of about 97% of mitochondrial ATP6 and ATP8 protein synthesis. It was also impairing the ATP synthesis levels about 50%, and we could measure co-localization of the fluorescent labeled RNA with the mitochondrial compartment using confocal microscopy. In another experiment, we aimed to deliver DNA via our vector system into the mitochondria. The DNA here was a single-stranded circular DNA of the bacteriophage M13 as a model. Has a size of more than 7,000 nucleotides, and this DNA was featured with RNA domains, with mitochondrial RNA domains. And each mitochondrial targeting vector here comprises already 8 of these RNA domains. So it means that here the vector on the right side, indeed, this DNA is featured with 40 single RNA domains suitable to help this vector being delivered into the mitochondria. And we can see here below, the more of this mitochondrial targeting domains we are employing, the better this single-stranded DNA is being delivered into the mitochondria. And the largest vector here has an overall size of more than 15,000 nucleotides. And it means also we haven't observed any upper size limitation yet, neither for the vector nor for its cargo. And could also here identify or measure co-localization of the fluorescent labeled vector with the mitochondrial compartment in the human cells. So in addition to using RNA and delivering the RNA with liposomal nanocarriers, we also explore using a DNA vector to deliver this mitochondrial targeting RNAs. And the vector we are favoring, the structure, is a dumbbell vector. This is the smallest possible genetic vector we can imagine. The dumbbell vector structure is shown here. It just comprises the gene of interest in the core, double-stranded DNA, including a promoter, a coding sequence, and a termination signal. So this could be the gene encoding for the mitochondrial targeting sequence. And the ends are then closed on both sides with single-stranded loops. So this is the smallest possible vector we can imagine, and the small size facilitates cellular, but also nuclear delivery. They are exonuclease resistant because they don't have any free ends. They are not immunotoxic and redosable. And very importantly, they are not silenced in primary cells, so there's no transgene silencing happening. They are non-integrating and considered safe. And via the loops, as indicated, we can conjugate these vectors with helper functions for target delivery or fluorophores. So we have attached, for example, cell-penetrating peptides, antibodies, small molecules, and aptamers can be attached. To give you an idea about the activity of these vectors, you see here on the left side the short-term kinetics from 10 minutes to 24 hours, and comparing the dumbbells with the plasmid DNA. The plasmid DNA is the black line, the dumbbells are the colored lines. You see that using lipofaction, the dumbbells are delivered much more efficiently into the target cells in vitro. Nuclear diffusion also is about 100 to 200-fold faster. It's a log scale here. Accordingly, the transcription from the dumbbell vector starts much earlier, and the dumbbells show a stronger phenotype in the targeted cell. More interesting even are certainly the expression profiles in human primary cells. As we can see here in the center, the plasmid-based expression is already silenced on day 3, whereas the dumbbell vectors here, there's no transgene silencing happening, and they show a normal expression profile over a couple of weeks, as we would expect in a tissue culture cell, but these are primary cells. And also the dumbbell vectors are significantly more stable as compared to plasmid or chemically modified RNA. Here, stability was measured in primary human T cells, and after 10 days, we can still detect between 50 and 90% of dumbbell vector DNA of the initially delivered dose, whereas the plasmid was degraded to a level of 3.5%, and also chemically modified RNA to levels lower than 10%. So one slide about the dumbbell technology, a bit more in detail. The dumbbells, if you compare our latest generation now with the conventional plasmid, the dumbbells show clearly more than 100-fold enhanced gene expression. And even compared with the previous state-of-the-art of a normal dumbbell, the recent generation is about 60-fold stronger with regard to gene expression. This is mainly two different features. Most important is certainly an active nuclear import signal, helping this vector being imported into the nucleus. We also optimize the structure of the dumbbells, as you can see on the right side, the conventional structure with a base paired core and the single-stranded loops. We included mismatches in the end here, rendering the ends more flexible, which facilitates nuclear delivery through the nuclear pore complex via facilitated nuclear diffusion. And again, this makes the dumbbells about 6-fold better with regard to gene expression as compared to a conventional dumbbell. How do we produce them now? We have invented 4 methods. I'm focusing on one method here today, the most interesting maybe. It's a 1 tube, 2 enzymes, 3-hour reaction, which does not require bacteria or cell lines. And it results in very pure and high yields of dumbbell vector DNA. You just need a template. This can be a plasmid or a PCR product, and then perform a PCR using chemically modified so-called gap primers, which have an abasic position here. So it means the polymerase will stop here, producing a PCR product, which has 5' overhangs. And these are designed in a way that they preform the shape of the dumbbell, positioning the 5' phosphate just in front of the 3' OH group on both sides, ready for ligation. So it's a PCR followed by ligation, and in 3 hours you can produce a dumbbell vector DNA. So to summarize this, we have here this new mitochondrial delivery vector system, which allows us to deliver RNA or DNA or both into the mitochondria. We managed to trigger the combinant mitochondrial gene expression. We managed to trigger more than 97% of knockdown of mitochondrial gene expression, and succeeded to deliver a single-stranded circular DNA of more than 7,000 nucleotides using a vector which comprises more than 15,000 nucleotides in total. This, and we know we can either deliver our sequences using liposomal nanocarriers exogenously into the target cells, and then the sequences will be targeted to the mitochondria, or alternatively deliver a DNA vector, for example, using liposomes or helper functions, and for example, a dumbbell vector, which then has to enter the nucleus. The RNA will be made and will be exported, but not translated into the cytoplasm. Instead, it will be targeted to the mitochondria and trigger its function. And no size limitation observed for this vector system yet. So currently, we are optimizing the technology. We are trying to we study this in lone cell lines, in cypress cell lines, in zero cells, and then hopefully also shortly in a mouse model, monitoring the expression of the restored proteins, and ideally a functional restoration of complex 1 function and visual preservation in the mouse model. So in conclusion, with this novel genetic vector, we aim to enable mitochondrial gene therapy in general and of lone disease in particular. Currently, we optimize the technology and aim to study this in a mouse model shortly. And this therapeutic approach shall prevent or restore loss of vision in lone patients and will be explored for targeting other optic neuropathies, including glaucoma. Later on, glaucoma is a much larger market, as you know, as a lone disease. I want to thank, first of all, my team, the current team, Rachel, Mei Ling, Derek, and Chen Xing, and also our alumni, mainly Kaur staff was involved in this work, our external collaborators, Professor Krausten, Trau, and Trounce, as well as the funding agencies. And thank you for your attention.
I don't know, this says that, Dr. Patzel, can this be a potential delivery vehicle for current anti-vascular endothelial growth factors? If yes, can we skip the injection?
I mean, the eye, you want to inhibit the vascular endothelial growth factor, yeah, right? So now there is an aptamer available, Macugen, which is binding to VEGF. So what you want to deliver in this case is an inhibitor, like an antisense molecule or an siRNA. And yes, we can. This can be considered. We can use, of course, either the RNA. RNA can obviously be delivered like an aptamer, but we can use the dumbbell vectors to express the small RNAs. We have made this already. So I'll show you one slide with a short expression kinetics. This was indeed in dumbbell vector expressing a small hairpin RNA, triggering a knockdown of a target gene. And these vectors, they can be as short as 130 base pairs if they are small RNA expressing, including the promoter. So very tiny vectors, indeed the smallest ever reported expression vectors with a very high stability, much more stable than the RNA alone. This is certainly something to be investigated, yes.
So I don't see any other questions here. And yes, it was indeed a very fascinating topic. And I know there could be a lot of scope for the concept and the technology in the near future.
Have
Yeah,
a good day
so,
and a good weekend ahead.